For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right, we are live for a really good show tonight, guys. I am pumped. We've got another must-watch lecture with Brother Sal. Today's lecture is going to be on protein probabilities. Now, this is part one in a series of lectures, guys. So get out your pens and papers because you're going to want to make some notes on this one. Also, we are going to make it interactive, as in we will be fielding some questions from the chat. Okay, as long as it pertains to the topic at hand, clarifying questions, anything in regards to the topic, of course. Quick announcements. We have Dr. Rob Stadler just confirmed will be back with us this week, Friday at 1 p.m. EST, 12 Central. So we're looking forward to that one. We're going to be talking about his book on evolution. And Wednesday, I myself will be debating on modern day debate. And we've got a couple other things to look forward to, guys, that I'm just in the midst of confirming, and then I'll announce it. So anyways, enough from me. I'm going to hand it over to Sal. Sal, I did mute you just in that moment because there was a bit of a background noise. I imagine it's probably fine now, but definitely unmute yourself before you speak. Sal, thank you so much for being here.
Thank you so much, Standing for Truth. And thank you all to the audience. Tonight, I'll be talking about protein probabilities. It can be sometimes difficult to navigate through this because there's a lot of terminology. So I'm hoping to make it at least somewhat interesting, but also hopefully the listeners who don't have a biology background can learn some of the terminology. So I'm going to share my screen, and I'm just going to go through my presentation. And if it turns out that I'm not communicating well, we're just gonna kind of ad lib and try to learn together, because this is a chance for me to try to learn also how to communicate the material better.
Yeah, don't even overthink it, Sal. You've been here a number of times for lectures, and they are always awesome. I've watched them a number of times. Like I said, these topics are so interesting. Your lectures are so educational that they oftentimes require multiple viewings just to get it all. So, and that's why I'll periodically take questions from the chat in case there is some clarification that's necessary. Sal, I did forget to mention to the chat, there is going to be an after show on Sal's channel immediately following this lecture. This lecture will probably go for about 60 to 90 minutes, including the question and answer. Then, guys, head over to Sal's channel for the after show.
Yes, I'm going to provide a link a little later, but I didn't want to keep the audience waiting. So we'll go through this, and when there's some downtime for announcements, I'll start providing a link to the after show. So the topic is understanding protein probabilities. Some of the probabilities that creationists and ID proponents have put forward over the years, they're getting just a tad obsolete just because we know so much more. And so this is an update on being able to understand the problems. I'm happy to report that the problems are far worse for abiogenesis and evolution than we imagined. The early work, a lot of creationists will cite work by Doug Axe, which is very fine work, but that's 20 years ago, and it only focused on one protein family, the beta-lactamase family of enzymes, which is a monomeric protein. And that's significant because that actually was a simpler protein than some of the ones we'll be studying this evening. Now, last night I was over there trying to break through the gates of hell there at the Saturday night Satan worship over at Speed of Sound's channel. And I was just typing in the chat. And I said, and you could see that in the chat, let me highlight it. I said, hidden Markov analysis show all proteins do not have common ancestry from a single gene. So a lot of times evolutionists keep saying, "Oh, there's common ancestry, common ancestry." I said, well, it's more subtle than that. There's no common ancestry in proteins. Do you agree or disagree? And of course, Speed of Sound says, "Why the hell would it be from a single gene?" And then Call Me Emo said, "Salvador Cordova, who claimed that they would or should all share a common ancestral gene?" And so I challenged them. So I said, "It's Speed of Sound. It shouldn't descend from a single gene. All the major gene families don't have a common ancestor, best as I can see. Agree or implicitly disagree?" And they responded some more. Call Me Emo said, "Call Me Emo." That name reminds me of Tick Me Elmo. Anyway, Call Me Emo said, "No one is claiming they were derived from the same ancestral sequence. You're arguing with yourself here," which I wasn't actually. But anyway, I responded, "So you don't believe in universal common ancestry of all genes?" And Emo said, "No, not all genes are derived from the same ancestral genes."
Awesome.
And Emo denied. So I said, "Emo, Call Me Emo denies universal common ancestry of all genes, proteins." So I said, "Thanks." Speed of Sound agreed. No universal common ancestry of genes." And so I just kind of rubbed it in. I said, "So you deny universal common ancestry of all genes and proteins too. Thanks." Okay,
you see
so
how they debunk themselves?
Uh,
you notice how they debunk themselves and admit, looks like indirectly, I don't even think they have realized it, Sal, but you've gotten them to admit a position contrary to universal common ancestry and what they hold to. And they look rather angry there too, Sal, while you look calm, confident, collected as usual.
yeah, you totally get it. They're saying, "Oh, you're strawmanning this, and why would you think something so stupid?" It's like, well, I actually, I actually don't think that they all descended from one gene. I just came to the realization of independent origins, and all the other guys on the channel were saying independent origins. You mean like an orchard? Okay, so I don't think they realize they're kind of giving away the store here. This is great. I don't even need to argue with them. So everything you're going to hear from this point on is just going to reinforce this. So this is one of those where they're not going to, they don't have any, to my knowledge, they don't have too many debunking videos of some of my lectures, like my one on fitness and hyalinase. So I don't see why they would want to debunk this one.
No,
they'll be creative, I'm sure.
We've got a full playlist with your lectures, and there's not even been an attempt. Maybe the best that they've done is straw man or misrepresent. But I love how you can get the little gems like you just did, just by asking them simple questions where we don't even realize it. They're promoting independent origins, which is what we believe in.
So there, you got something to start with now. You can bash them over the head with that now, because this is where it's going to begin. So a lot of times we work with the organismal common ancestry, but I said, let's just start on something a little bit more basic with proteins. Now we basically started to saw the universal tree of life kind of at the root. Maybe we've not cut the whole tree trunk off, but we've damaged it some with this one. And creationists, when I put this question before creationists, they said, "Yeah, actually, that's a pretty good question, Sal." I'll relate how I came up with this. So the lack of UCA is universal common ancestor. So this is universal common ancestor of proteins. So this relates to the difficulty of de novo protein emergence. Many de novo proteins had to basically poof out of nowhere with no ancestor. No macroevolution. This also implies there's no macroevolution from one evolution of one protein to another. That is, from one family to another. So a member of one family can't macroevolve to another. So these guys basically have just kind of given the store away. So when I said the de novo proteins had to poof out of nowhere, so if you had independent origins, it's like, well, where did it come from? Some sort of random soup. And I really like this quote by James Tour. He said, "Origin of life research is retarded." That was just at Syracuse talk. And of course, this is one of my favorite ones. You're telling me this is, there's a chance. So let me stop sharing this. And I'm going to put in a video. Do you have any comments while I try to get this video going?
I guess just for the audience, as the lecture proceeds, if there's any clarifying questions or even any skeptics or critics hiding out in the live chat, if you have any questions or comments as well pertaining to the topic, though, of protein probabilities, tag me and we will stop at certain points and relay them to Sal. So no, that's really it from me, Sal. I can whenever you're ready for me to screen share the clip, I can go ahead.
So this is just a little bit of comic relief. I need a little bit of comic relief every now and then just to clear my head.
Well, they say laughter is the best medicine.
Yes.
And we did that with the last lecture you were here on the worthless definition of evolution. We had a lot of comic relief for the first hour, thanks to our good friend Speed of Sound.
Speed of Sound.
That's not what I meant. What I meant was, I like you, Mary. I like you a lot. I want to ask you a question straight out, flat out. I want you to give me the honest answer. What do you think the chances are of a guy like you and a girl like me ending up together?
Well, Lloyd, that's difficult to say. We really don't...
Hit me with it. Just give it to me straight. I came a long way just to see you, Mary. Just least you can do is level with me. What are my chances?
Not good.
You mean not good like 1 out of 100?
I'd say more like 1 out of 1,000,000.
So you're telling me there's a chance? Yeah!
Classic. Sal, you know what that makes me think? I feel like that is the skeptics and critics that we deal with all the time that are incapable of actual sophisticated rebuttals. I feel like that is, let's take Speed. Speed always gives us a good laugh. That's like him asking his evolutionary naturalists and the PhDs working in the field, what are the chances? Please tell me, what are the chances of abiogenesis being true? Hit me with it. I don't want a designer. And then, you know, one in a trillion is really what it would be. But to them, Sal, it's the same reaction. So you're telling me there's a chance, right? Regardless of how low that chance is for something like protein evolution, universal common ancestry, abiogenesis. So that's a great visual that is there for comic relief, but it speaks so... there's strong meaning to the evolutionist, the naturalistic camp. So, Sal, I think you're on mute, brother, if you wanted to say something. But that was my thoughts on the clip.
Thank you. It looks like I had the wrong slideshow, and I'm gonna stop screen sharing, and I'm gonna bring up another screen share. Apologies for the technical difficulties. We had Murphy's Law in play earlier. So I'm going to try to reattempt my screen share here.
I'm going to point out, Lena Powell says, brothers, there is a chance in the multiverse. Sal, I love how you pointed out yesterday that the multiverse that they look to, Noah's flood is probably real in one of those trillion universes. So I liked how you pointed that out. Go ahead, Sal. Go ahead.
I think I got my slides back in order.
There we
Okay,
go.
great.
You got
so
it.
Let's move on. just a little bit about me. I see some questions in the chat. I'm going to rely on USFT to kind of filter them out. I appreciate all the questions, and maybe we could queue them up just so we could allow the flow. I'm not trying to ignore. All the questions are interesting. This is the Evolutionary Genomics Lab. This is the staff of the Evolutionary Genomics Lab at the National Institutes of Health. And pictured here in the middle, if you could see my cursor, is Eugene Koonin. He's one of the top evolutionary biologists on the planet. He's the head of this group. This was my professor here, who is in Koonin's group at the FAAS graduate school at the NIH. And so I have studied some evolutionary biology, not a lot. And some of the stuff that I will talk about tonight is inspired by that. There is here a paper published in Nature, which is the top prestigious scientific journal. And you'll see a name that might be familiar to some of you, Joe DeWeese. He is a creationist who got published in the world's top scientific journal. And he gave his testimony in the movie Is Genesis History? And I had the privilege of being his co-author on a publication in biochemistry, molecular biology in the FASEB journal, which is some sort of federation of experimental biologists. This was actually a diagram that appeared in our poster session at that conference. And I'm happy to say I'm the one who built that diagram. And I'll actually talk a little bit about this if we have time. And so here again is Joe DeWeese's work in the prestigious scientific journal Nature. So that's just a little bit about me. So just going back to the main point, the lack of a universal common ancestor in proteins, it relates to the difficulty of de novo protein emergence. And as I mentioned, this would imply many proteins had to poof out of nowhere with no common ancestor. And there's also no macroevolution from one protein to another. That would be major groups. There'll be microevolution, which you can cover. And just to repeat, if you reject universal common ancestry for proteins or genes, why stop there? How about no universal common ancestry for eukaryotes and prokaryotes, et cetera, or anything else? How about no universal common ancestry, period? Now, I call the protein universal common ancestor PUCA. I pronounce it PUCA. So if there's no PUCA, then we have to explain the independent origin of protein families. This would imply miracles are needed for a scenario where there is no PUCA. And so let's continue on. So a technique I use is argument by contradiction. That is, we assume for the sake of argument something is true. So we assume a false statement is true to show that it's false. So I'm going to do that a lot. I am a young earth creationist, but I'm going to be making a lot of arguments that, for the sake of argument, assume common ancestry. And you can tell when I'm switching out of that paradigm into creationism, but I'm just going to assume for the sake of argument for a lot of the talk in universal common ancestry, it will show PUCA doesn't exist. Also, if we assume universal common ancestry, this whole thing about human similarity, it doesn't solve the problem of de novo origin of major protein families. So sometimes when I argue with evolutionists and they're talking about pseudogenes and the ape-human similarity, I'll just say, this stuff really doesn't solve some of your other major problems. You better be thinking about solving the major problems before you start straining at these little gnats, because you're letting elephants and camels through. And this whole problem of the universal common ancestor of proteins is a big deal. And I will try to argue tonight that evolutionary theory needs statistical miracles to get it going. One thing that is often done, and it's misleading. I'm reluctant to use the word dishonest. but it's very misleading. A tornado can pass through a junkyard, and it can make paperweights, primitive hammers, and clubs, but it can't make a 747 jetliner. So let's say a tornado passes through a junkyard. It can make a primitive hammer, but it's not going to be able to make a jackhammer that has all these gizmos and needs electricity or whatever, or some engine to get it going. So it's not very honest to take one example where we take a random polypeptide and show that it can catalyze some reaction and then say, hey, well, look, we've made a functional protein. That's very misleading. It's not legitimate. It's not appropriate. So I will actually argue that creationists should not be saying, and I'm a little bit of an outlier here, we should not be saying there's no chance a random polypeptide can make a functional protein. It can.
Oh, Sal, it looks like we may have you frozen, which has happened before and isn't an issue. The best way to fix it may be to jump out of the stream and then come back in and kind of reload it up. But other than this freezing incident, it's been flowing good. Audio's been good. Visual has been good. So guys in the chat, let's just wait for Sal to come back in. And let's see how everybody is doing. Otangelo, good to see ya. He says Coonan believes in the multiverse. Yes, they will believe in anything as long as it's not God. Alec Cox, SFB Mob, Dessel Drace, good to see you. Glad you're enjoying the content. Godzilla Freak says, no God, infinite, undetectable universes on the other hand. Yes, I think they've been watching too many Marvel movies. I'm a superhero fan, but at least I realize that, those are fiction. Evolutionist seems to watch these movies and take them as truth and science. Otangelo, yes. Those are some crazy odds, my friend. Those are some crazy odds. So anyways, for anyone who just got here, this week, we are going to have Dr. Rob Stadler back on this Friday talking about his book on evolution. It's going to be a great discussion. You guys are not going to want to miss it. Those not familiar with Dr. Stadler, check out the Professor Dave debunking we did and share that around. Share that around. Make sure Professor Dave knows that is out there. I know he does know it's out there. Says apparently, according to him, not worth responding to. In other words, that means he is incapable of refuting the facts. Part of me thinks he regrets putting out a video attacking Dr. Tour and attempting to defend chemical evolution. So Alec Cox says, what's up, SFT? Not a lot. Having a good day. Been a busy week. But it's been fun. It's been fun. We want to put out as much content as possible for you guys. Chris Keith says, can't wait for the Dr. Stadler return. Yeah, we got a lot more good discussions and interviews ready for you. Brother Sal from Evidence and Reason says, this is Sal on backup computer restarting internet. No problem. Take your time, brother. Take your time. This was the moment to engage with the audience. There he is. Sal, we miss you. It's not the same without you, Sal.
Thanks. Thank you to the audience for forbearing. So
No
where
problem.
where were we? I was talking about collagen. Did we get that far?
Yeah, right there. The tornado through a junkyard slide, I believe, is what you were on. But
Okay.
yeah,
Let me cover that again. Thank you all. Thank the Lord that I was able to reconnect. So I was saying that a tornado can pass through a junkyard. It can make paperweights and primitive hammers and clubs, but it can't make 747 jetliners. And I need to point something out here because evolutionists will use kind of a dishonest argument, misleading. I mean, I rarely try to call them dishonest or liars as much as I'd want to do that. So let me just say it's very misleading and inappropriate. So if we had a tornado passing through a junkyard and it created all this junk, more junk, and we said, hey, let's grab a piece here from, some of the junk lying around and say, hey, this is a nice paperweight. And if I said, see, tornadoes can make something functional, that wouldn't be a very legitimate argument, would it? But some of these examples of novel proteins being generated randomly, it kind of is in line with that, where they can take random polypeptides and they can find some reaction that it can catalyze. It's misleading to suggest that this can evolve into some of the other kinds of proteins that we'll explore today. It's really not very honest. Also, too, yes, sometimes a protein can be subject to some random mutation, and it can actually change its function in a way that might lead it to be able to do a few other things, but not a radical change. So let me just move on. Do you have any comments on that? So now let's go to collagen. Can you hear me okay, SFT?
Yes, your audio sounds great.
All right.
And no specific questions or anything yet from the audience on the topic. So just continue on. Everything's flowing good, Sal.
Okay, so there is this beauty product that one can buy at Walgreens. It's Collagen Bodybuilder. Collagen is a real protein. It accounts for 20 to 30% of the human body by mass. The protein weight, the dry protein weight, 20 to 35 to 30%. So collagen's a real protein. And just to cover some basics, there is a collagen gene on the DNA, and it gets transcribed to an mRNA for collagen that becomes a protein. So this is kind of basic molecular biology. So if we have kind of a random DNA coding sequence, it'll make a random protein. So just to point that out, I'm not going to be dealing too much with the DNA and RNA side. We'll just assume, we'll just focus on the protein, and we'll think about the random statistics of how this protein might form. So this is the spelling. Can you see it? The fonts may be a little small. This is the spelling of the collagen 1 alpha 1 paralog in humans. And it's 1,464 amino acids long. Now, there is a subtle, do you see a pattern there? I'm going to expand it. Do you see a pattern? You'll see it there.
Yes.
So these are the amino acids, and each letter represents an individual amino acid. So M is for methionine, F is for phenylalanine, S is for serine, et cetera, et cetera. The G here is glycine. So you can see there are only about 2 protein families that have just these glaring patterns that are just so obvious. It's definitely non-random. Some evolutionists will argue, well, there's some mechanisms that can do tandem repeat, so this is no big deal. That's a really superficial argument, and we don't need to go into that right now. But basically, the first problem is this is not an exact repeat for the letters that are in between, and plus there are other problems with that hypothesis, which I'll point out. So this is just a superficial look at the collagen. If we go to the UniProt database and give a little bit more annotation of the parts, I'm going to give you kind of a parts list of that collagen. So this is the same. What I'm going to show is the same protein here, but then with some annotations that show the pieces of what's going on. And you could see there are a lot of parts that are actually identified as important. All right. So this is giving an idea of the complexity of the protein. We're not even looking at the 3D shape if you just kind of take the pieces together. Lots of proteins, especially in humans and higher level eukaryotes, have complexity like this, just rich in complexity. So this is kind of a visualization of the important parts. One can see that, like if you had a frameshift mutation, this protein's just totally gone. The spelling's just destroyed. if we insert even a letter here, that repeating G pattern is going to be destroyed. That is an important property that makes collagen have its physical properties. So I'm going to note something here. I identified what's called the excision region. That means this is going to get snipped off as the protein. So when it goes out of the ribosome, this protein is going to go through more processing. It's going to have the start of the protein snipped off, and then the end of the protein snipped off. And these parts have particular names and they have particular functions. We don't have to go. We don't have to go into all the detail of what they do. In fact, we're still trying to learn what all these pieces do. But this is a good spot to pause. Are there any questions?
There we go. Sorry, I was unmuted. Yes, let's see. So 2 questions so far. The first one was more so based on our abiogenesis talk earlier. Desseldrace asks, he's curious of your opinion on Nick Lane's account of abiogenesis in The Vital Question. Are you familiar with that, Sal?
Not at all. And that is a very good question. The person to ask would be Rob Stadler this Friday.
Ah, yes.
He's the expert.
Well, that's what I said in the number of times we've had you, Dr. Stadler, Ryan Hayes, the skeptics. If they think that, if they're confident with abiogenesis, email me your best challenging questions, and we'll have Dr. Stadler address them. So,
let me say this. Can I add one thing? If Nick Lane really thinks he has a solution, he should claim the $10 million prize.
Yeah, exactly.
All right. So money speaks here. Okay. So there'll be a panel of experts evaluating it. There was a earlier one that was offered for $1 million that was headed by David Abel, and the panel of judges were atheist Nobel laureates, and no one claimed that prize. So, my opinion doesn't really count that much compared to the experts who are adjudicating the $10 million prize. So
right.
I'm sorry. I thank you very much for the question, Desseldrace. I'm sorry I couldn't give you something more substantive than that.
Look at the
speaking of sounds there, you can ask him if he thinks all proteins descended from a common ancestor.
Yeah, I told him that he was the star of the show for the first 15 or so minutes at the beginning of this stream. So, Speed, when you get a chance, make sure you check that out. It was a lot of fun. I guess his question is, can proteins fold around sodium ions?
I don't know. I haven't studied the sodium ion channel. But the potassium ion channel, it actually doesn't want the sodium ion it doesn't want the potassium ion channel to converge around the sodium ion. That's how come the potassium ion will fit through the potassium channel. And the reason I know that is that's one of the proteins, if we have time, that I want to cover. So
awesome. Well, that's all I've got so far, Brother Sal.
All righty.
I'm enjoying it. We're all loving it. So if you want to continue, go ahead.
So for anyone that wants to know where I got this graph from, I did it myself. One can go to the Uniprot website and type in this identifier for the collagen 1A1 human collagen, and you'll get all the coordinates for everything. You can build a diagram like this from scratch. So this isn't, even though I created the diagram, the data came from public domain sources. So notice again here, this part on the top, and then this part at the bottom. And I'm going to just forward to an illustration here. You could see here that it's going to snip the pieces off. And I'll explain a little bit why there are 3. You actually need 3 collagen polypeptides to make a tropocollagen. And we'll go into that a little bit more. But you can see how this snips off. And let me see if I can illustrate that. So you can see how the part that's snipped off here corresponds to this part. The middle part here corresponds to this part in the tropocollagen. And then this NC1 domain that it's called corresponds to this part here. There is a protein that actually needs an enzyme to be able, oh, there it is, procollagen peptidases. We'll cleave these things off. So what does this mean? For collagen, let me move forward a little bit. So we have the procollagen that comes out of the ribosome. You actually need 2 copies of the one paralog and then a copy of another gene that comes from another gene. So you need 2 genes to make this collagen, where you have 3 strands. And then the 3 strands create the procollagen, and then that becomes the tropocollagen. The tropocollagen then becomes part of a collagen fibril, and that becomes the type 1 collagen fiber. So there are a couple things to note. We actually need an enzyme. So some dumb evolutionist, sorry, he's someone I really don't like. He's on another forum. He was saying it's so easy to make that. You just make a repeat. And I just wanted to say, you buffoon, that won't work, because you need another enzyme to start finishing the process. It has to snip those pieces out. And I'm not even done with this. There's also another enzyme. Do you see these green things here? Those are chemical modifications called post-translational modifications. You need yet another enzyme to add these chemicals there, otherwise it's not going to fold right. So you need at least 2 pre-existing enzymes to make this work, and you need another gene that's on the genome to make it work. So you need, and we can go into that if someone has questions. So what does this mean? We can't just throw a protein together in isolation. If we just had this protein without all that other machinery, it's just not going to work. It's not going to serve any function. So this whole thing about, when evolutionists will say they got some random polypeptide to catalyze a reaction, that's to extrapolate it to the systems that involve something like collagen. That's totally illegitimate, because we need several simultaneous the existing parts to make this work. So now a little bit of cell basics. We have here, this is a eukaryotic cell. So there's some elements that eukaryotic cell shares with a prokaryotic cell. Eukaryotic cells are like the cells that are in humans, and prokaryotic are the ones like in bacteria. And it has what is called a plasma membrane surrounding it. The prokaryotes have something similar. They have a cell membrane, just a little bit slightly different architecture. What sets the eukaryotes apart are these membrane-bound organelles like the nucleus. It also has a membrane. It has also a lipid bilayer membrane. So there's membranes here made of fat, lipids. The proper term is lipids. And then lipids here. It's almost, the architecture is very similar to almost like a cell. In fact, the mitochondria, do they have it? Yeah, it almost looks like a prokaryotic cell here. It also has a membrane, and that's very important. So this leads to transmembrane proteins, and I'm gonna let me stop. I'm gonna stop sharing my screen here. Let's see if I can bring up a video.
Sal.
Yes.
Speed, our good friend, he just said, just watch the beginning. He says, all proteins did not descend from one gene.
Thank you, Speed. No universal common ancestor for genes. Thank you.
He says we're doing great work, and he's one step closer to being converted. So he's being a real sweetheart in the chat. So we're working on you, Speed. We're working on you. I can sense that you're slowly coming to the realization that naturalism just isn't the answer. So I'm sorry to interrupt you on that, Sal. Go ahead.
No, that gave me. You see, you're giving me a chance to actually get all my computer stuff set. So it's good that we have a little bit of relief here. I'm going to share my Chrome tab, and I hope this doesn't blow up. This video is going to be pretty technical. So just to forewarn you, it's 4.5 minutes long. There's a lot of technical terms. And actually, I think I got the wrong video. So I just realized this. I need to put a different video in. We'll get this right.
Anthony, you take your time. I'll just engage with the audience, Sal. You take your time. Anthony's just saying, Speed, come over to our side. Abiogenesis is a colossal failure. I'm pretty sure that Speed had a light bulb go off. He had one of those realization moments a couple of weeks ago when he tried to refute. We had Dr. Sarfati on, PhD chemist, and he just tore apart abiogenesis. And Speed tried to do a rebuttal to it, and he got about 1.5 minutes into it, and you could just hear it in his voice that he was thinking, oh man, I can't answer this. So he shut it off immediately. So the point is, Sal, we want to bring these skeptics to the truth and ultimately to Christ. So okay, looks like we got your. I killed some time there, Sal.
Yeah,
I see your
thank
video.
you. So this is going to be really technical. So just kind of the parts where they throw terminology that you're not familiar with. For those that are not molecular biologists, including myself, I'm not a molecular biologist. Just forbear with it, because there'll be parts that should be understandable, and I might even pause the video and we could just talk about it a little bit in case I see that there could be some confusion. So we'll just go with it.
Membrane proteins are those proteins that are either a part of or interact with biological membranes. They make up around 1/3 of human proteins and give different kinds of membranes their unique properties. They help with both facilitated diffusion and active transport, connect cells together, participate in signal transduction, and act as markers for cell identification. Proteins are what carry out most of the specific functions of membranes, so the amount and types of proteins vary between different membranes. Membranes can be up to 75% protein by mass. Membrane proteins come in 2 flavors, integral or intrinsic, and peripheral or extrinsic. Integral membrane proteins are a permanent.
I'm just pausing this a little bit. For those of you who watched the James Tour lecture, he was really tearing into Jack Shostak, who was saying, oh, we found a way to make a kind of like, for lack of a better term, I call it a protocell or whatever he called it. He said, oh, we can make lipid bilayers. They form spontaneously. And that was being advertised as one step closer to solving the problem of the origin of life. Rob Stadler and James Tour, but Rob Stadler summarized it really well. He said, well, Jack Shostak basically invented a nice coffin for nothing to ever come to life. Because without these proteins that are on the membrane, particularly the ones that allow things to go in and out, the thing is a sealed coffin. And James Tour called him out. It didn't matter that Jack Shostak is a Nobel laureate, because he should be called out on something like that. It's like, this is not, you can't be advertising something that is a sealed coffin that will kill life as a step closer toward origin of life. That's very misleading, and he should be called out on it. So this is what this is dealing with. So this membrane layer here, it could either be on an organelle or in a eukaryote or on the cell surface. So I'll just continue.
Permanent part of the membrane, while peripheral proteins are only transiently associated with either the membrane or integral proteins, where these associations are hydrophobic, electrostatic, or other non-covalent interactions. There are several different kinds of integral proteins. Integral monotopic proteins are attached to only one of the 2 leaflets of phospholipids making up the membrane, and they don't span across it. There are also transmembrane proteins and lipid-anchored proteins. Transmembrane proteins are those that span the lipid bilayer, and can be bitopic, spanning across the membrane once, or polytopic, spanning across it more than once. Lipid-anchored proteins are those which are covalently attached to lipids embedded in the lipid bilayer. For example, GPI, or glycosyl phosphatidylinositol, is a glycolipid that gets attached to a protein C terminus during post-translational modification. It acts as an anchor for proteins to the outer leaflet of the plasma membrane. Both integral and peripheral proteins can be post-translationally modified. There can be addition of fatty acids, diacylglycerol, prenyl chains, or GPI. Recall that cellular membranes are made up of a phospholipid bilayer, which consists of 2 leaflets of phospholipids. These phospholipids have polar heads which are hydrophilic, or water-loving, and nonpolar fatty acyl tails that are hydrophobic, or water-hating. Polar substances like to interact with other polar substances, and nonpolar substances hang out with other nonpolar substances. This really attests to the power of hydrogen bonding. Water molecules want to interact so badly with their polar buddies that anything nonpolar getting in the way of their hydrogen bonding results in decreased entropy. The result is what's called the hydrophobic effect. This is why phospholipids in water will spontaneously form lipid bilayers. These bilayers minimize contact between polar and nonpolar molecules, maximize hydrogen bonding, and maximize entropy. This is also why transmembrane proteins are amphipathic, which means that they have regions which are hydrophilic and regions which are hydrophobic. The hydrophilic regions are exposed to water on either side of the membrane, while the hydrophobic bits are happily interacting with the hydrophobic tails of lipid molecules in the interior of the bilayer. As a result, transmembrane proteins are stuck permanently in the cell membrane and are very hard to isolate. To get them out, you need to add detergent, which is amphipathic and will disrupt the lipid bilayer. There are 2 basic types of transmembrane proteins, alpha helical proteins and beta barrel proteins. Note that while helix bundle proteins are found in all types of biological membranes, beta barrel proteins are only found in the outer membranes of gram-negative bacteria, mitochondria, and chloroplasts, evidence that contributes to the endosymbiotic theory, in which eukaryotic cells acquired these organelles through the ingestion of prokaryotes. Transmembrane protein structure can be predicted using a hydropathy plot, which has hydrophobicity index on the y-axis and amino acid number on the x-axis. The amino acids making up a protein are localized according to polarity within its final structure, in such a way that the polar amino acids face the outside aqueous solutions, and the nonpolar amino acids are adjacent to the lipid bilayer. Transmembrane proteins can be classified by topology, which is based on the position of N and C termini, as well as start transfer and stop transfer sequences. For example, type 1 is a single transmembrane pass with the N terminus on the extracellular side of the membrane. Type 2 is also a single transmembrane pass, but the N terminus is on the cytosolic side of the membrane. How these different topologies come about will be the topic of another video. Often, transmembrane proteins function as gateways, allowing specific substances to pass across the membrane. They can undergo conformational changes as they do this. They might participate in facilitated or active transport. Facilitated transport is spontaneous passive transport of substances via transmembrane proteins. Active transport, however, requires energy. Active transport may be necessary, for instance, if a substance is being carried across the membrane against its chemical or electrical gradient. As a final note, in animal cells, most transmembrane proteins are glycosylated. These sugar residues are always present on the non-cytosolic leaflet of the membrane. As a result, the cell surface is covered in carbohydrates, which form what's called the cell coat. If you enjoyed this video, like and subscribe.
So the point was just trying to introduce some of the ideas of the importance of transmembrane proteins. Without them, there's no life. Without, you'd be dead real fast if they weren't there. And there's no hope for abiogenesis. So I'm going to show some videos then, and then let me try to resume my slideshow here. See if I can resume it.
Oh, I see it now, Sal. I see it. Boom, you're good to go. You're good to go.
Okay, so I'm going to probably what I'm going to do is I'm just going to I probably won't be able to get to the topoisomerase and the zinc fingers, because as I was developing this, I said I really need to cover the transmembrane proteins. So if you had any questions here, this is a class of proteins that's not explored very much or talked about. One of the reasons is it's actually very hard to study them, very difficult to study them. As they were mentioning, you have to use very strong detergents to break apart the cells and the membranes to be able to get a hold of them. It's not an easy thing to study transmembrane proteins. But James Tour, in his talk criticizing the origin of life, and I love what he said, origin of life research is retarded. Nobel Prize winners like Jack Szostak showing these little spherical things that have lipid bilayers with no transmembrane proteins. That's very misleading, as I mentioned earlier. And so transmembrane proteins probably have to be there from the very beginning if we're talking about cellular life. You want to talk about something else? Yeah, sure. Maybe you could build another kind of replicator, but we are interested in the origin of cellular life. So how can we have cellular life without transmembrane proteins? So when we covered collagen earlier, and I'm showing a picture here, there is what's called the localization signal. And that helps it go through these membranes here and go to the right location in the cell. It's called the localization signal. So if we're talking about protein probabilities, one of the features of a protein is that many of them need localization signals so they know where to go in the cell, or at least they will announce to the rest of the cellular machinery that is there to transport cells where it has to go. So you need a localization signal. So that's something that is non-random. So these experiments where they just take random polypeptides and say, look, it can catalyze a reaction, that's misleading, because real proteins, like in a eukaryotic context especially, need these localization signals. They're like postal codes. And I'm going to show how some of that works. So collagen has a postal code here. That's localization signal. And we have a transmembrane protein that has localization signals here, highlighted in yellow. And I'm going to show how that is processed and incorporated into the cell membrane. This is a potassium channel. This is a potassium channel. So I'm going to stop sharing and I'm going to grab another video. And we're getting close to the end, and I've had too much fun.
Take your time, Sal. This is too much fun. People are loving it. Logical, plausible, probable says, where are these membranes in the official, quote unquote, evolution timeline?
Where are these transmembrane proteins? Oh, I've got one. Okay, so we had DNA's Diet Coke. DNA's like Diet Coke. I've got another quote. Okay, I got another one. It's not quite as good. It's Jackson Wheat. I'm gonna nail Jackson Wheat on this one. Okay, so I'm gonna show you how difficult it is to stitch in a potassium channel. So this one's going to show just kind of a general thing of how a transmembrane protein is stitched into a membrane. So back to logical probables, probables question, where does it fit in the timeline? It's like, yeah, that's a good question that's never been answered. And I'm going to show some of the complexity. So it's not just the problem of just throwing up a random polypeptide. It has to be able to be recognized. It needs proper signaling to enable it to be incorporated and stitched into into the membrane. And I believe this video is going to show that. Oh yeah, I got it. So let me, I'm going to share my screen here. I mean, I'm not giving hard numbers, but I mean, I'm just trying to give a feel for you saw that postal code. That's a pretty long sequence.
Oh, yeah.
It's just not going to happen randomly.
No.
For it to be recognized. And so what makes it improbable is you need both that signal to be there, that sequence to be there, and more importantly, you need a machine that can recognize that signal. That's what makes it improbable. I mean, you don't need a very long string to start to make this astronomical. Both the sequence in the protein that has that postal code, and then the other protein that comes along and recognizes that postal code. That's a highly improbable thing to be able to build machines like that in the cellular context. So, I mean, if someone wants to really work through the numbers, I invite them to. That'd be a great exercise. But it's like at this point, I'd like, as I was saying last night to Derek, at some point, Noah's flood actually looks far more promising.
Right. I loved how you put it that way, and I loved how you phrased that, because what we know about the improbable nature of these types of evolutionary scenarios that they have to hope, dream, and imagine occurred, long ago and far away. It makes believing in the global flood of Noah very, very easy in comparison. So I really like the way you put that. And I see your video here, Sal, and we'll add to the stream.
All righty. I'll mute my mic.
The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are two separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. Two kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling a sewing machine. As they are being synthesized by a mechanism resembling a sewing machine.
That's
Okay,
amazing.
now I need to. I'm just going to pause it here because this is such a great picture. This is why I get sometimes mad when they say, "Oh, look, we were able to get some little protein to evolve." I'm just like, that is so illegitimate, because the problem that needs to be solved is to be able to evolve a system that has so many interacting parts that are so critical. Like, just in reviewing this video, we need the signal recognition protein. We need another protein called the signal recognition receptor. We need a translocon channel, which is this blue thing here. We need the signal peptidase, which is going to cut pieces off. And then we need a plug when it's all done. And in this case, this protein has these multiple signals here. So you have basically a sequence that's red and then another that's orange that has to correspond. And this has to, this is in the context, this is just to get it to stitch through. That we're not even talking about it being functional. So what's represented here is a protein that's incorporated in the endoplasmic plasmic reticulum. There's similar principles in putting this at the cell membrane, the outer part of the cell, or any other organelle. So now let me show any questions or comments, and we're going to try to close it out in 15 minutes, if that's okay.
Yeah,
because I'm also running out of gas.
No problem. I mean, I'm loving it. We're loving it. Whenever you want to finish, let us know. I mean, this is part one, so whenever you feel comfortable finishing, and we're going to be heading over to, I'm going to have people head over to your after show anyways. The comment that sums it up was, where was it? It was SFB Mod. Joel said, look at all, I'm trying to find it so I can put it on the screen, but I think I lost it. He sarcastically said, look at all that random evolution. Isn't that funny that they could say all of this for no reason at all, by chance? I mean, it's amazing one could conclude such a thing as compared to worship the creator. So I shared your screen again, Sal.
Thank you. So you saw in that video how it's stitching through and recognizing parts, and the parts that span the membrane. What I've highlighted in yellow, and again, I got this from the Uniprot website where we get the coordinates. These are the parts that go through the membrane here that stitch through. Isn't that cool? And so just imagine then when it's stitching through, it's recognizing these signals here, and they're proteins that have to recognize this. So now I'm going to show how this thing works. I have a video of this is the potassium channel. I don't know that this is the same one in this video. Let me see if I could find it. Oh no, don't tell me I don't have it.
Sal, you're getting us.
No, I think I have it here. Oh, I have it here. Praise God, I have it here. Hang on, because this is just too good not to show. Let's see if I could put it in there. Let me see if I'm going to load it, and you're going to see how this baby works. Oh yeah, I got it going. Hang on. It's coming. So now I'm going to share my, let me share my screen again. The reason I'm having to do this is we found out StreamYard really likes Chrome tabs. So I'm flipping back between the Chrome tab and the PowerPoint presentation. It's kind of difficult to embed it. So that's a StreamYard issue. So I'm going to show how a potassium channel works.
Perfect. No, you're doing it good. Whatever you're doing, it's everything's going smooth. Anthony Marie says, Sal, thumbs up. Excellent. Yeah, this has been great. We've still got 30 people in the chat, so take your time. Sal,
Okay.
get through what you need to.
Thank
This
you.
has been fun.
So I showed that sequence, the potassium ion channel sequence earlier, where I highlighted in yellow. And I was pointing out that's the thing that gets stitched in between. Now we're going to see how this thing actually works. What ends up happening is the gene has to make 4 copies of that protein. The technical term is polypeptide. And so this will cover how this thing works.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
Now I'm going to point out something. This is why it suggests there had to be foresight to build this, because the first thing is when this protein is made, it doesn't work if it's only 1 copy. It's made with the anticipation that the 4 copies will somehow connect to each other, and it's stitched through by all that machinery. And so it anticipates that it's going to fit here, and it's showing that it's only going to it uses some really clever physics to enable it. In fact, it uses that word perfect. And this is one thing I think is really important. Let me just try to pause it. Oh, I'm sorry. Let me try to get it to pause at the right spot. Well, anyway, I'm kind of failing at that. There it is. It says perfect fit. It's very hard. You know, we think that, oh yeah, if we have to fit something, we like say we want to tap out a hole so we can put a screw through it. We just kind of have to mill it through until it's just right. At the atomic level, you can't do stuff like that. If you want to build a port, you have to use some really clever molecular engineering. And this fit is so precise, it is sub-angstrom. It's fractions of an atom precise. Is there anyone is there any machine shop that can make something that is precise to precision of atoms? A lot of biochemists would have a very hard time building a channel like this from scratch if they didn't know how it was done in the cell. So there's tremendously ingenious engineering here going on. And so this, in my debate with Ron Garrett, I was trying to point out this is the better idea of what fitness really means. It means there, at least for the potassium channel, there's only one fit. you can't mutate this and make it better. The only way this thing will change is that it will be neutral at best and most likely a damage to the system. So any questions? Are you there, SFT?
Yes, this is amazing. The chat's loving it. My question would be, actually, before I ask a question, all I'm thinking while watching this and listening to you is that it really is science fiction, what's going on in the molecular level, of the cell. And the more that we discover, the more that it shouts a designer and a creator. This stuff's amazing. It makes me wonder, Sal, what is the evolutionists, what is their best explanation for this stuff? I mean, are they even attempting it? Like, what's their arguments in the face of this data?
They use these, what I call these mumbo jumbo phylogenetic arguments that aren't mechanistic explanations. So they could say, oh yeah, this is related to that because there's sequence similarity, therefore it evolves. And there are problems with that, which I can't go into right here because phylogeny is another topic. But the thing is that I try to point out to them, you're assuming the thing you're trying to prove, you're assuming just because you have a phylogeny that there's some process that prevents the creature from dying in the process of evolving this protein. And it's an open question for me personally. Probably someone knows the answer, whether there's any organism that can live without a potassium channel. That this may be critical. I tried to look into Craig Ventnor's minimal cell to see if there was a potassium channel there. I don't know for a fact that if it's there, but there's certainly certain proteins that are transmembrane proteins that if it's not there, the creature's just dead. And so James Tour is right to call Jack Szostak out on Szostak's work because he didn't address these. These are serious problems. It's like, how are you going to have cellular life without these transmembrane proteins to let stuff go in and out? So when James Tour covered that in his talk, it's like, yeah, this is really cool. And I was really glad he had the courage to show that, because then that made it easier for me to say, well, hey, if he says this is bad science and this sort of origin of life research by even a Nobel laureate is retarded, I'm like, okay, I have the courage now to say that it's retarded. That they think they're actually advancing the origin of life question, and they're not. They're not at all, because they have to solve real problems like that. Being able to just create a lipid bilayer is not solving the problem. In fact, as Rob Stadler pointed out, it's a dead end. You actually show how you can kill a cell. He did a very good job of showing how you could kill a cell, and yet it's represented as an advancement. And I find that very troubling, and James Tour does as well. So now that we see the complexity of transmembrane proteins, now it's my chance to call Jackson Wheat out on something.
Yes, please do. He has brought this upon himself, Sal, so call him out.
So let me do a screen share, and this is kind of the this will be kind of maybe a good stopping point. So I was in my first debate. It was really more of a conversation. We were just talking, and he was very kind. Someone said, "You got to debate Jackson Wheat," and so we just had a conversation. I said, "Let's not debate. Let's just all we're going to do is say what we believe and why." And I was telling Jackson Wheat, I said, it's so hard to evolve a eukaryote because it has all these membrane-bound organelles. And so what good is it to have an organelle there if it doesn't have transmembrane proteins? If it doesn't have transmembrane proteins, that's just, basically a dead organelle. And so Jackson Wheat said, well, like a leaky membrane, for instance, then stuff could go in and out. And that's not how a membrane works. It's meant to prevent stuff going in and out randomly. So this is the last thing you want. That's why you need transmembrane proteins. And James Tour pointed the problem out for a cell in general, but it applies also to the membrane-bound organelles in a eukaryotic cell. So I have to give Jackson Wheat a Diet Coke award for that statement. That's how an evolutionary biologist will solve it. They think in terms of phylogenetic quote unquote reconstructions. They don't think mechanistically.
Well, in other words, they don't think empirically. We're looking for an empirical answer to this. And what you said earlier about foresight, foreknowledge, that those forward-thinking mechanisms that we see in the cell, in the genetics, requires a forward thinker. But evolutionists then, the naturalist that rejects the forward thinker, they have to apply a mind to evolutionary processes. And I also like how you pointed out the fact that it's much easier to break something, to kill something, than to make something better and to create something from scratch. Like, for example, the examples you're showing here, which is just fascinating. I mean, this is, it's amazing stuff, Sal. You're doing great work, brother.
Thanks. Just for completeness, the video was for a prokaryotic cell. The potassium channel I showed was for humans. It's just just for completeness, in case someone wants to ding me on that. I couldn't get a video for that particular channel, but it's the same idea. And in case Jackson is wondering where I got it, it's about 30 minutes into our first debate in his Hangouts on Air. So let's see.
Oh, perfect. I'm glad that you're showing it too. Now we can see the.
Yeah. So I mean, there he is. Okay. So then, just for the record, so about 30 minutes in, about 32 minutes in, that's where he says he could just leak in and out. And I was just like, dude, have you not had cellular biology?
Apparently not.
You're kind of being brainwashed. I mean, you need to go back. I mean, that would be just like, oh man, you could tell I tried to be very polite and just kind of let him talk. But, I wasn't going to nail him on that. But then he said some negative comments about me after the show, which I didn't appreciate. When I debated Dr. Dan, he was saying I was melting down. I said, okay, all right, Mr. Nice Guy is kind of ended here. I'm going to call you out on junk that you said specifically, and that was garbage. And this is,
well, you have to earn respect. And if he's going to say those things about you, the thing about Jackson Wheat is he's kind of on the sidelines now. He's kind of irrelevant. He didn't look good at all in not only your discussion with him, but also with Dr. Kevin Anderson. Like you felt bad for Jackson during that debate, put it that way. And here's the problem, because a lot of these critics like Jackson, they don't really think for themselves in terms of their arguments against our position, as in they're kind of just repeating arguments that they've heard in blogs.
Yeah, they're
like
talking
that.
points.
So, talking points, exactly.
They're not thinking like, you see, my background is in engineering, and it's just natural to say, well, how did it work in between? How did this change without breaking the system? It's just a natural thing to ask. They don't ask these questions. So I'm sorry to pounce on him a little bit, because he was really my first debate ever, and that's how I got to be on. That was my introduction to the blog sphere, so to speak, or the debate sphere. And but I just, I finally said, this needs to be called out. He is repeating evolutionary talking points, and all of the papers on transmembrane evolution are just phylogenetic reconstructions, which aren't mechanistic explanations. They don't seem to get it.
Yeah. Historical reconstructions are only as good as like the empirical data that we have in the present, for example, like what you're talking about, this irreducible complexity, that which evolutionary processes cannot explain. That means those historical reconstructions, those phylogenetic reconstructions, like you said, it's just essentially a story, a reconstruction of the past where they're assuming universal common ancestry. But what you're showing, Sal, demonstrates that universal common ancestry, it's not a justified assumption. I mean, because they've got, you said it yourself, so their best argument, Sal, is to not really explain it. They just shift to their phylogenetic reconstructions. You know, that doesn't seem like a good answer to me. That's just my opinion, though. It doesn't seem like they have a real good answer for it.
Well, this may just briefly to anticipate kind of maybe the other segments, the other parts, the other presentations in the series. When I showed those sequences of collagen where you could see that very clear repeating G pattern, the collagen, and then I showed another sequence for the potassium ion channel, you could see the spelling is very, very different. It's not just minor changes. The collagen for humans was 1,464 amino acids. The potassium channel was under like 500. It's just unreasonable to think they had a common ancestor. So going back to the whole thing of why even evolutionists would agree with me, they reject universal common ancestry for proteins. It's just too blatantly obvious. If you throw all these proteins, well, you know, what you can do is you could take proteins and throw them into a sequence alignment program like MEGA or some what they call hidden Markov models, and it can try to estimate if they're related and how closely. Well, when you take all the sequences and throw it in the computer, all the sequences in our databases, it's not going to find a common ancestor. And so hopefully by actually showing you the sequences, a little bit of it, you could see why that the lack of universal common ancestry, it really does hold water, especially if the evolutionists will admit it, albeit reluctantly. They don't talk about it. So I'm making them talk about it. Next time you ask them, you confront them, you just ask them, well, do you believe in universal common ancestry of all proteins? And so now I've hopefully given you some ammunition, because that's a question we haven't been putting on the table. We need to confront them with that. And then say, if you're rejecting universal common ancestry there, why stop there? You can reject it pretty much everywhere. So anyway, SFT, I'm gonna try to put a link in the chat for the after show.
And well said there, Brother Sal. I was on mute interacting with the chat. So this has been awesome. I mean, this hour and a half has flown by. I'm already looking forward to part 2. So you just let me know. You name the date. We'll let the part 1 get some views and allow some people to study it. And then in the coming weeks, we'll schedule part 2. I got to say, Sal, we are pumped. We're excited that you are as active as you have been, because we've been having a lot of fun. You really tear down universal common ancestry and naturalism. And that has been proven just through your series of lectures and videos you've done with us. There's been no serious response to it. And that actually reminds me, on Dr. Sanford and Dr. Tour's interview, I really liked, Sal, how Dr. Sanford pointed out that really the only responses to his work have been on the internet through blogs and keyboard warriors. Go ahead.
My link isn't going through. I'm posting as evidence and reasons because that's my website.
Yeah, I got to make you moderator now.
That's a different name, and I tried to put it, and it wouldn't let my link go through.
I'm doing it. I'm fixing it right now.
Or I could put it.
You're good to go. You can post it now, Sal. I made you mod.
So I'm gonna try to go over there right now and try to get my StreamYard going. Hopefully it won't blow up.
Well, I gotta say, guys, if you have any questions or anything regarding the topic at hand, I'm saying later on when you guys watch it, email me. I can pass them on to Sal as well, but we will, or you can just gather questions that we can maybe answer a few before, when we do part 2, say in a week or so, we can do a little Q&A prior. But yeah, I'm looking forward to it. I want to thank the chat. We had a good audience tonight, some good comments, good conversation and questions. Sal, as well as always, thank you for your time. We're gonna get people subscribed to your channel, and we're gonna get your stuff out there, Sal, because as far as I'm concerned, this stuff is a must see. You know, people need to know about this. So we're going to get it out there. Sal, God bless.
God bless. I'll
Okay.
see some of you all at the after show. Take care.
Okay, guys. SFT.